Hello, welcome, thanks for joining. Today I want to talk about an approach we developed in our lab for multimodal analysis of cell-free DNA whole genome sequencing data for pediatric cancers with low mutational burden. My name is Peneder. I'm a bioinformatics PhD student in the Tommaso Lab for epigenome-based precision medicine at the St. Anna Children's Cancer Research Institute in beautiful Vienna, Austria. I want to start by giving a brief introduction into liquid biopsies. In patients with cancer, the tumor releases cell-free DNA fragments into the bloodstream during secretion or during necrosis or apoptosis. Liquid biopsies refers to the analysis of these cell-free DNA fragments. Now, the key challenge for the use of liquid biopsies in oncology is to distinguish DNA fragments that are derived from the tumor from those that are derived from healthy cells. And usually this is done by the detection of somatic mutations. But we are working with pediatric tumors with low mutational burden. So we are taking a slightly different approach, as you see in a second. But first, I want to give a brief motivation of why we care about really bringing liquid biopsies into clinical practice. Solid pediatric tumors are a challenge for clinical monitoring because they require invasive biopsies or imaging. They cannot be performed every time the patient presents at the clinic. So clinicians are faced with limited information. If they had access to high resolution data, this could improve treatment outcomes, for example, by allowing earlier detection of relapse than is possible with traditional methods. In order to analyze our dataset, we used a fragmentomic approach. And this refers to the fact that while the cell-free DNA is degraded, released, it gets degraded in a non-random fashion. The cleavage occurs preferentially at stretches of DNA that are not protected by nucleosomes. And therefore, the fragmentation patterns allow inferences of the epigenetic states of the source cells. We analyze these fragmentation patterns in a paired and whole genome sequencing data of our liquid biopsy cohort, which is a cohort of healthy controls, Ewing sarcoma samples, which is a rare pediatric sarcoma type, and patients with other sarcomas. We focused our analysis on the coverage at regions of interest, and these regions of interest can be, for example, DNA hypersensitivity sites or enhancer peaks or other regions that are epigenetically active in your tissue of interest. And what we find is that if these regions are active, we see a depletion of nucleosomes around these regions and therefore a depletion of sequence fragments. Therefore, we can observe a drop in coverage if the cells that give rise to the cell-free DNA are epigenetically active in these regions of interest. In order to quantify this phenomenon, we developed a tool called LIQUORICE, which detects tissue and cancer-specific epigenetic signatures in whole genome sequencing data from liquid biopsies. LIQUORICE takes as input a set of tissue or cancer-specific regulatory regions, for example, DNA hypersensitivity sites, and a set of mapped reads of your sample of interest. It then calculates a bias-corrected composite signature of the coverage around these regions of interest. LIQUORICE works as follows. It splits every region into bins and then calculates the coverage and the number of bias factors for each bin. These bias factors, for example, the GC content, are important because they influence the coverage without being related to the epigenetic properties that we actually care about. So in the second step, we correct these associations between bias factors and coverage. In the third step, we aggregate our coverage signal over all regions in the region set in order to obtain a more robust signal. And finally, we fit 3 Gaussian functions and an intercept to this coverage drop, which allows us to extract metrics such as the area over the curve or the total depth of the dip, which you can then use to make comparisons between samples. And when we do this for our cohort of Ewing sarcoma patients, patients with other sarcomas and healthy controls, and when we use a set of Ewing sarcoma specific DNA hypersensitivity sites that we derived from the literature, we find the following. Healthy controls and non-Ewing sarcoma samples show a flat profile with even a small peak coming up, whereas many of our Ewing sarcoma samples show a drastic drop in coverage around the center of these sites, and especially those for which we have confirmed genetic tumor evidence as well. So this is nice. As a positive control, I included a set of universally accessible DNase hypersensitivity sites, and you can see there's virtually no difference between the groups here. The Ewing sarcoma specific coverage signal as calculated by LIQUORICE is a very quantitative measure of the tumor content. So it's highly correlated with the percentage of tumor-derived cell-free DNA that we infer from genetic metrics. But this is true only for our Ewing sarcoma samples. You can see the non-Ewing sarcoma samples show almost no correlation, which highlights the specificity of this approach. I'd like to mention that LIQUORICE is also applicable to healthy tissues, such as blood or liver, and it's also useful for other tumors. So we have applied it to rhabdomyosarcoma as well, for example. So this really generalizes beyond the application to Ewing sarcoma, which makes this, I think, a more interesting tool. Finally, we took LIQUORICE, our LIQUORICE measurements, and integrated it with other mutation-independent fragment-based metrics that I unfortunately don't have time to go into. And we use these as features to train machine learning classifiers to distinguish Ewing sarcoma samples from healthy controls. And as you can see here in these receiver operator curves, these metrics perform quite well in distinguishing the samples, with the best feature reaching an area under the receiver operator curve of up to 0.97. We can also take these features to distinguish Ewing sarcoma samples from other sarcomas. And as you can appreciate, our LIQUORICE-based approach clearly works best here, which is an area under the receiver operator curve of 0.92 and clearly outperforms the other metrics. This brings me already to the end. I just want to mention that we are currently looking for a bioinformatics master or PhD student to join us. So if you're interested, head over to our website, tomasolab.org, for more details, or directly get in touch with me. And thanks a lot for your attention. I'm happy to discuss any questions, feedback, comments, just get in touch. You can also check out LIQUORICE on GitHub, drop me an email, read our article, which I posted in the poster channel, or head over to my talk. So just get in contact. I'm happy to hear from you. Thanks a lot. Bye-bye.